Okay, so the next kind of mutation is the chromosomal mutation, and chromosomal mutations, as we have already seen, they are the mutations with respect to the structure of the chromosome or the number of chromosomes. So, chromosomal mutations produce more pronounced effects than a single gene mutation, and they are inherited once they occur and may take place by several methods. Okay, and they are otherwise referred as chromosomal aberrations. There are two kinds of they are inherited in two ways actually. One is the structural changes in chromosome, and other is numerical changes of chromosome. Okay, structural changes in a chromosome could be due to changes in the number of genes. For example, deletion or deficiency due to loss of genes from a chromosome. So, a segment of a chromosome may be lost, and this segment may contain one or more genes. Okay. Then duplication due to addition of a gene to a chromosome. So, duplication here mainly refer to a particular segment of chromosome may get duplicated or may get copied and then added to the same to the chromosome. So, that is a duplication. Okay. You can see over here with example. So, deletion, what happened is, this is the actual chromosome and a fragment, a small fragment has been removed. Okay. And what happened to the chromosome? It has decreased in length. Okay. Now, duplication, what is happening? This is the actual chromosome length, and this is the segment which is getting duplicated, and another copy is added to the same chromosome, and this is known as a duplication. Okay. So, what happened to the chromosomal length? It increased. It increased with by the size of the duplicated chromosome segment. Okay. So, this is what is duplication referred to. Changes in the number of genes in a chromosome can also occur as a result of rearrangement of genes, and this rearrangement of genes, it could be by inversion or translocation. Okay. So, here in this figure you can see inversion. Okay. Inversion, it is a reversal of the normal sequence of genes. Here you can see this is the normal gene, normal chromosome with the gene A, B, C, D, E in the sequence A to E in this way. Okay. After inversion, what happens is the C and E gene had got inversed. Okay. So, the sequence have changed from A, B, E, D, C. Okay. These are the two arms. Okay. In this arm, there are, in the normal case it was C, D, E sequence, but because of the inversion in the position of the gene, it had got E, D, C arrangement. Okay. So, here the chromosome breaks at these points and then joins again, but it is turned 180 degree. That is what happens. Okay. So, inversion, it is actually a reversal of the normal sequence of genes. Okay. It is actually CDE, instead it becomes EDC. So, there is a breakage and then it turns 180 degree and then it joins again. This is what happens. Okay. Now, the next one is the translocation, rearrangement with respect to translocation. Here what happens is two chromosomes are coming into play, where there is an exchange of blocks of genes or a segment of chromosome between two non-homologous chromosomes. I hope you remember what is a non-homologous chromosome. Homologous chromosome is the what you call sister chromosomes, isn't it? The two chromosomes will be of equal length with what you call centromere at the same position, right? But these are non-homologous chromosomes. They do not have any with respect to size or length of the arm or the position of the centromere, they do not have any similarity. Okay. So, these are the non-homologous chromosomes, and then there is an exchange of the chromosomal material between the non-homologous chromosomes. So, what happens is there is a complete change in the chromosomal length, the position of genes, etc. So, all these major changes contribute to the increasing viability by variation by changing the position of genes, and this is what is referred as the position effect in a chromosome. Okay. The position of genes, it is getting changed, isn't it? And that is why it is known as a position effect. The first one was point mutation. Okay. And this means that variations are produced not only by changes in the structure of gene, but also by its position in association with other genes. That is, with respect to other genes, the position of a particular gene, if it changes, it can also cross a mutation. Okay. And this can be can result in variations, right? The next one we have is the numerical changes of chromosome. changes in the number of chromosomes, and it could be the what you call heteroploidy or genomatic mutations. Here it is caused by the addition or loss of one or more whole chromosomes or sets of chromosome. In the structural change in chromosome, here the whole chromosome is not getting affected, only set of genes are getting removed or added, or position is getting affected. But in the numerical changes, what happens is one or more whole chromosomes may be added or lost from a genomic set, or sets of chromosomes may be added or deleted. Okay, so that is what happens. So such variation in the number of chromosomes, it is known as heteroploidy, okay, or genomatic mutation. Okay, it is referred as heteroploidy or genomatic mutations, right? Okay. Now it can be classified into two, that is euploidy and aneuploidy. Okay, I hope yeah, here you can see with respect to numerical changes in chromosomes, the genomic mutation can be of two types, euploidy and aneuploidy. Okay, euploidy, it is a change in the entire set of chromosomes, that is a whole genome is added or deleted. Okay, it is further divided into monoploidy, or it is otherwise known as haploidy, then polyploidy. Okay, haploidy, here it is the basic set where each chromosome is represented only once, right, and this is the condition in the gametes. I hope you remember when you may have studied mutation, or sorry, what you call mitosis and meiosis, the gametes were represented as one N, that is a haploidic condition. Okay, and when it got fertilized by the sperm, after fertilization the zygote is formed, and the zygote had the genetic composition of 2N, isn't it? That is a diploidy nature. Okay, so here we can say that there is a single set, basic set, and it is represented only once, right, and this is the condition in the gametes of animals, and even some of the body cells of many plants. Normal animal cells have diploid condition, it is 2N. Okay, that is a 2 haploid cells or 2 genomes. So a genome will have only haploidy condition. I hope it is clear. Another condition is the polyploidy, addition of one or more haploid sets of chromosomes to the normal diploid set is known as polyploidy. Okay, so it could be 3 sets. When it is 3 sets, it is 3N. Okay, and it is referred as triploidy. When it is 4N, 4 sets of genome, 4 sets of haploid chromosomes may be present, and it is referred as a tetraploid. Okay, likewise 5 set, if it is 5N, pentaploid, 6 sets, 6N, or it is referred as a hexaploid. Okay, and it can go on. Okay, depending upon the number of genomic set being added to the normal chromosome number. Fine. Increase in number of chromosomes leads to greater increase in the size of body. So body size increases as the number of chromosome increases. Okay, along with that, it shows vigorous growth. Such an organism give better yield, and they show more adaptive value to the possessor. Okay, polyploidy seems to have been important in the evolution of plants. Okay, very rarely in animals it is found, but in plants we can see plenty of examples for polyploidy, successive polyploidy. Now, with respect to aneuploidy, aneuploidy refers to loss or gain of one or more chromosomes. Here, one or more number of chromosome is added. Against in euploidy, the whole set. Okay, this is a single set. Okay, the whole set of chromosome is added or erased. Okay, so that is euploidy. Aneuploidy is one or more number of chromosome is added or it is lost from the normal chromosomal set. Okay, it is again it could be different types. There are monosomy, you have nullisomy, and you have the polysomy. Okay, monosomy is where the one chromosome is lost from a normal set. So the normal set is always 2N, diploid condition. Okay, so it is 2N minus 1, that is monosomy, right. Nullisomy is a whole pair, chromosome pair is lost, okay, and that is 2n minus 2. A whole chromosome pair is lost, okay. Then the polysomy means addition of one or more chromosomes. It could be 2n plus 1 or 2n plus 2, likewise, okay. So, addition of one or more chromosomes to the normal diploid set is what is referred as a polysomy, okay. I hope it is clear, right. Aneuploidy results in genic imbalance, and hence development of the organism is abnormal in most of the cases. You may have learned about Klinefelter syndrome, Down syndrome, isn't it? In all these cases, what happens? One or more chromosomes are lost, isn't it? X0 condition, XXY condition. So, one chromosome is added, 2n plus 1, right? X0 condition, there is one chromosome is lost, okay. So, I hope here the examples cited are for the aneuploidy, okay. So, aneuploidy, it results in genic imbalance, and this development of the organism are getting disturbed in usual cases, right. So, chromosomal aberrations are distinctly harmful, but some like polyploidy have an important role, especially in the case of cultivated plants like wheat, and appear to play an important part in the process of evolution through the origin of species, right. So, that is with respect to the mutations, chromosomal mutations. So, we saw genetic what you call gene mutations, the characteristic features of gene mutations, then chromosome mutations, the different types of the chromosome mutations, okay. Now, the importance of mutations in evolution. I hope this can be dealt in the next presentation, okay. Fine, thank you.